Well, welcome everybody. Once again, we're having a seminar that's jointly sponsored, this time by the NCI Physical Sciences Oncology Center and also by the Molecular Biophysics Training Grant. Our speaker today is Wilma Olson from Rutgers. Wilma received her PhD with Paul Flory, who's really, I would say, the original hero of polymer physics and polymer physical chemistry at Stanford. She then went on to work with Charles Cantor, is that right? Yeah, who's really the hero of nucleic acid biophysical chemistry and polymer chemistry. And then since then, Wilma's been an independent professor at Rutgers, where she's taught us most of what we currently understand about nucleic acid topology, and then more recently has hugely enriched our understanding of how proteins see nucleic acids and of the sequence-dependent nucleic acid structures and dynamics that allow for indirect readout and nucleosome positioning and lots of other hugely important phenomena. So we're really lucky to have Wilma here today, she'll talk today mainly, I think, about DNA mechanics and nucleosomes out of this larger context of the body of her work. So welcome, Wilma.
Thank you very much, John. And I shall also tell you a story about how I first met John. I think it was at a Gordon conference, and he was working with Buzz Baldwin, a professor at Stanford whom I had known. And so, of course, I was very interested in what he's been doing over the years and have been following all kinds of exciting work done on nucleosomes from his lab. So I'm really delighted to be here, but I really admire an awful lot of work that's going on here in structural biology and in biological physics. So I'm very glad to have the opportunity to speak here. I'm going to start by telling you a little bit about the people with whom I'm working. Currently, there are 2 postdocs in the lab whom I'm working with, and we're trying to understand various aspects of DNA supercoiling, including some simulations of nucleosome-bound DNA. And we're doing that work because we're working on a project in conjunction with Vasily Studitsky, and it builds upon work that his former student, Yuri Polikhanov, did in trying to learn about long-range interactions on chromatin fragments. But today, all I'm going to talk about is how we're sort of gearing up to do these kinds of things and how we're working slowly by looking at high-resolution nucleosome structures to learn things and develop models where we can treat larger systems. A lot of the work that we've done at Rutgers has built upon the work of former students and graduate students who are in a lighter shade of gray, including one undergraduate, David Mccandlish, who worked with me through a summer math program. He was a Swarthmore student, and I'll tell you more about what he did because he brought a very fresh perspective on what we're doing. And then I work a lot with Victor Zhurkin at the NIH, and some of the work we did involving David Mccandlish was also done with Misha Tolstorukov, who's now working at Harvard in the medical school. And we're very fortunate to have had NIH funding to do this work. So just to give you a sense of the talk, it'll be in 6 parts. There'll be an introductory part that tells you a little bit about some of the different way we've thought about nucleosomes. And then I'll tell you just a tiny bit about mechanics of DNA and how we've learned things about that, and how we can use derived information from high-resolution structures to estimate costs of deforming arbitrary sequences along arbitrary pathways. And then lately, we've been collecting a lot of information. There are now, in the 30s, 35, 36, 38 nucleosome crystal structures that are out there in the public databases. And so we've been looking at the collective information. And in looking at the collective information, we've been trying to study the proteins as well as the DNA, and have found that thinking of the nucleosome as a cylinder has really advanced our thinking. And then with that kind of information, we're working on these models where we can study nucleosome communication and work with Vasily's group. So you get a sense of what's going on. So here's our different perspective. This is the same old perspective that everybody knows, that the nucleosomes are these protein DNA assemblies that are so important for the packaging of DNA in eukaryotic cells, but also very important in controlling what's expressed, because where the nucleosomes are may occlude what's expressed, or where the nucleosomes are may enhance the binding of another protein that helps remove the DNA come off of the nucleosome. So there's a lot of excitement in the field in trying to think about that, and we're trying to think about it in terms of the perspectives of molecules so that you might be able to make predictive results. This is a kind of color-coded image. This is not the best resolved structure. This is just the first nucleosome structure that was solved by Carolyn Luger in Tim Richmond's lab, where there's 146 base pairs of DNA wrapped a little more than 1.5 turns around a core of 8 proteins, which from this point of the talk, where it's very DNA perspective, it's just purple stuff in the middle, and we're worrying about the bases. But toward the end, we're going to worry about the purple stuff in the middle. Now, again, the same old view, but again, looking at the perspective that people have taken in the past. What was early noted when the crystal structures were determined was that there were very key contacts made between arginines roughly every turn of the helix. And these were sites where the DNA minor groove was getting narrower. So the structure wasn't a perfect helix. It was bent around, and some of these strands were getting narrower. Also, in this particular image, we're trying to emphasize that these interactions were with the sugar phosphate backbone, really not with the bases. And a lot of what we understand about many proteins that interact with DNA that require particular sequences, their interactions with the bases. But the nucleosomes have to bind everything, so their contacts are primarily with what's common to everything, the sugars and the phosphates. So these arginines are shown in yellow that are making the contacts. And then you'll see red dots. And those red dots are basically points that are on the centers of the base pairs. And one of the things that you can notice as you go around the structure, and this was noted early on, that, here we're going along, and then there's a very sharp turn. And so what's happening, and these sharp turns are occurring in the vicinity of where these arginines are binding. So the DNA wasn't wrapping smoothly like a slinky around a beautiful cylinder, but it was wrapping, and at points where it was touching these cationic amino acids, there would be sharp bends in the structure. And when you have this kind of system where most of the interactions are involving the phosphates and the sugars, it's really hard to understand why certain sequences are positioned on nucleic acids. And so that, and considering interesting work that came here from Northwestern, the 601 sequence, for example, why in the world does that sequence bind so tightly to nucleosomes? So we started, and this really was the result of work with this student, David Mccandlish, and we started looking at nucleosomes from a different and a simpler perspective. So this is the same old view of the nucleosome. Here we've drawn a dot, and that dot is actually the line, the superhelical axis around which this DNA is wrapping. Computationally, what we do is we find a line which basically optimizes the distances so that they're all relatively fixed between the centers of the base pairs. And then here, the first half of the DNA on the nucleosome is shown with larger spheres, and the second half with a thinner line, which you can only see in part, and maybe a little bit toward the end, just to distract, so you're not distracted by the busyness of the whole structure. And then at these points, these are the sites where you can clearly see that the DNA is bending, very sharp bends, for example, and they're colored differently. And they're colored differently because there are two kinds of bends. In one case, where the bend is blue, the minor groove is getting narrow. And in one case, where the bend is pink, the major groove is getting a little bit narrower than what it would have been in the double helix. And then these points are labeled with a number 0 corresponding to this pseudo-symmetry point in the structure called the dyad. And then points like this, minus 3.5, means that you are 3.5 helical turns from the dyad. So from the black to the pink is 1 helical turn. From the black to the first blue is about a half a helical turn. So here's a half, 1.5, 2.5, 3.5 helical turns. Or this pink is at 1, 2, 3, 4, or 5 helical turns away from it. And then one will assign, and this started with the first structure from Luger et al., to assign numbers if you're preceding the dyad as you enter the structure. These numbers are negative with respect to the dyad, and on the other side, they're positive. But one of the things we did, which was a result of other things that I'll describe to you, is that we said, well, let's look at this structure, because we learned that we should look at the structure from a different visual perspective. This is the same old thing, but it's just rotated by about 45 degrees. And so these are those bends that are emphasized in this view. But every time you see the bend, you see little zigzags in the structure. And that was something that hadn't really had much attention paid to in the past. Now, these are the two kinds of bends that are going on. There's one kind of bend that I mentioned where the minor groove gets closer together. This is a schematic of two base pairs. And what's happening is that the edge of the base that faces the minor groove, they just get closer together. And the edge that is facing the major groove gets further apart. This is a side view of this kind of narrowing of the minor groove side and widening of the major groove side. The pink kind of bends are ones where the minor groove, which is on this side, is getting wider, and the major groove is getting narrower. But then these are these views where we look at it from a different perspective. Instead of looking down the structure, let's rotate it. And what you see is that those there's a kind of dislocation of the helix axis on these points, which is related to that change in the jogs that we were seeing in the structure. And in one case, the change in the axis is occurring at these points that are half integral turns away from the structure. And the slide is something that looks like this, which is arbitrarily assigned some positive number. Well, the number is the magnitude of the variation. And in the other case, this kind of variation of the bases, the shearing of the bases, is in the opposite sense, in a negative sense. So you have these two kinds of bends, the one kind of bend where the minor groove is getting narrower, and a kind of dislocation that's slightly different from the one where the major groove is getting narrower, and the dislocation is in the opposite sense. And this is just a kind of map of these two variables, the bending and the shearing variable. And we were looking at points such as this one at minus 3.5 and the one at minus 5. So we were looking at 3.5 where this number was greater than 2 and the bending angle was around minus 18. And we were looking at one around here where the bending angle was around 18 and the shearing parameter was around minus 1. Skip that. And one of the things I should point out is that these two little types of bends there's nothing very unusual about them. One where the bend is positive, which means that the major groove is narrowing and the slide is negative, is the kind of deformation that you see at every step in the aDNA helical structure. And of course, you can get from bDNA to aDNA by removing water. It's just in our case, these things are happening at isolated points. And of course, this kind of thing would change a 10-fold structure to an 11-fold structure. And the minor groove, which is highlighted by these darkened atoms, becomes very wide. Actually, my text is wrong. It's just, it says narrowed major groove. And that says narrowed minor groove. So the major groove, you can't even see it anymore. And here, for example, the kind of bend that occurs in the opposite sense was a form of DNA discovered 10 years after the Watson-Crick model called the C form. You get it by adding lithium to AT-rich sequences. And the minor groove narrows. And you get 9 residues per turn rather than 10. So these kinds of things in the nucleosome are the same principles that were seen in the earliest nucleic acid structures. Now this is the work that David Mccandlish, the undergraduate who came to our lab, did. He was a math major, and we were looking at nucleosomes. He said, oh, those are pretty. How are they put together? And so he wrote a little computer program where he was looking at all this variation of parameters and seeing, oh yeah, this is up and this is down. What if I just made the bend angle 0, this roll angle 0 everywhere? Or what if I just made the slide 0? He said, what are those numbers in ordinary DNA, bDNA? And roll is 0 and slide is 0. And so he did a series of computations where he generated 3D structures. So he was thrilled by this one. He made roll 0, and as we all expected, the DNA straightened out, because we all knew that that was what was contributing to the bending of the DNA. If he picked the one called tilt, the structure changed a little. It went from this orange thing, or from the white thing to the orange thing. But he was thrilled, and then on top of it, he learned how to make a movie, and he has a web page, and you can see this thing coming off. So this was a great summer for him. He wasn't as thrilled by this result, but we really were, which was, here's the plain old DNA, and it has some shift and some slide that are non-zero. And if you make the one parameter shift 0, nothing really happens. But if you make slide 0, that superhelix flattens to a structure that just wraps around itself. And so this kind of undergraduate investigation was the first thing that we saw, that slide somehow was contributing to the pitch of the nucleosome. And if we didn't have those slides, we wouldn't be getting a superhelical structure. Before that, most people thought about superhelices the way mathematicians do. They said, well, what's a helix? Why, it's just a circle, but there's an increase in the displacement of the atoms from the plane. And they thought again about it like a mathematician. And mathematicians put little coordinate frames called Serre-Frenet frames around a curve. And the way you get from a circle to a superhelix is you just introduce little twists at all these Serre-Frenet frames. So everyone for 40 years or more had been saying that, oh, well, what's happening on a superhelix is because you're introducing twisting at the same time that you're introducing bending. And it is known that twisting and bending are coupled in DNA, but people had just ignored this stuff, and what the literature was saying was, oh yeah, those things, they're just fitting the DNA up against the protein. But this kind of very simple undergraduate project said, maybe there's something more to it. And so this is an example of a side view of the nucleosome. And what we've done is we've picked out steps, one at the dyad, one that's at position -4, and one that's at position +4. So you have 80 residues, essentially a full turn of the superhelix. And these are points where there is very large variation in slide, -0.9, -1, -0.7. And all of these things are occurring. The slide move is this side-to-side move of the bases, and the side-to-side move of the bases is occurring in the same direction as the superhelical axis. This is one of those stereo views. So if you look at it cross-eyed, or if you put your eyes far apart, if you look at it cross-eyed, it's the wrong-handedness. If you can see it, with your left eye on the left and your right eye on the right, you'll see the left-handed axis. But that's just a kind of thing. But we can compute this kind of stuff. So we can say, well, there's 3 kinds of moves. There's these 2 kinds of shears in the plane, and then there's the spacing between base pairs. And so, we can look at every little base pair step, and we can say how much of the pitch was due to the shift, or how much was due to the spacing of the bases. And what we found is that as we went along the chain from 0 to 146 nucleotides, we would just collectively incorporate the pitch. And that if we were worrying about the spacing between base pairs or this other in-plane translation, it really wasn't contributing to the pitch. And when we looked at it, it really looked a lot like that first image that I showed you where we looked at the nucleosome from a different perspective. That is, we could see these jogs in the structure. And so these jogs in the structure where there are big steps occur where either there's this major groove narrowing and shearing that might be of the order of 1 angstrom versus the minor groove narrowing where the shearing of the bases might be the order of 2 angstroms. And so we had this kind of picture of the nucleosome where it looks more like a staircase than a ramp. And then here are just little bits of a real parts of the nucleosome where you can see these helical fragments. And so you can see these dislocations, which are essentially corresponding to these schematized stair steps. And here's a positive translation versus a negative translation. And that's important too, because these types of bends occur at half integrals of the structure, half a turn. And you know that when you look at a helix, half a turn away, the part of the structure that was in the front is in the back. A shearing in a negative sense at one position versus a positive sense at one position will dislocate the axes, this way or that way. But if you make the shearing, maybe a negative move here, and then half a turn away you make a positive move here, it's all going in the same direction. And so it's this alternation of bending with alternation of shearing that's contributing to the whole pitch. So it's taking advantage of the helix and this capability of DNA to not deform too far away from each other, to go toward an A-like DNA, to go toward a C-like DNA, but for this to occur in a kind of incremental fashion. So now some mechanics. So we were talking about the parameters called roll and slide. There are 4 other types of parameters called rigid body parameters that relate one base pair to another. This is just basic physics. If you have an object, you have another object, there's 3 translations that you take and 3 rotational moves that you take, and then the 2 objects can coincide. And so these are just popular names used in the structural biology community. But these 3 of the 6 parameters are the ones that show most variation in high-resolution structures. So here I'm showing you roll from around a couple hundred structures, no nucleosomes. And these are roll angles at steps that are G followed by G on one strand, C followed by C on the other strand. And so what it is we'll go look at some structure and we'll say, there's a GG step. What's the roll angle? And we'll collect it. And go to another structure. There's a GG step and collect it. And when you do enough of it, you get histograms and you see that there's a distribution of what this roll angle is around some number 5, 10 degrees. If you go to steps like AATT, you get a very different kind of distribution. The distribution is centered around 0, as if these steps, their bending isn't very different from what ideal canonical BDNA is doing. Similarly, there's differences in the twisting, not quite as obvious as these differences, and differences in the shearing. The shearing is closer to 0 in the case of the AAs and more displaced from it in the case of GGs. So this kind of variation suggested that there may be something intrinsic to the sequence that maybe proteins like the histones in the nucleosome could be taking advantage of. This tries to make the point more strongly. These are the same old bases, C followed by A on one strand, T followed by G on the other, as pyrimidine-purine steps. But here, it's just reversed to a purine-pyrimidine, A followed by C, G followed by T. And these are collections of the bend angle that we were talking about, roll, and the shearing parameter we were talking about, slide, in 239 structures, none of which are nucleosomes. And this is observations of the data of CATG steps in all kinds of structures. And these are the AC-GC data. That is, these are the values that you see in roll and slide in these structures. And they're also, we're careful, these are only AC-GT steps that are in the middle of the structure, and they don't have any weird things next to them, no metals nearby, no modified bases. So it's very vanilla DNA. But you can see that these data are very, very different. They're spread all over the place with the pyrimidine purine, and they're very clustered in the case of the purine pyrimidine. So you can do statistical analyses, and you can find mean values of these parameters. You can find covariance, and you can find sort of the direction in which these parameters vary. It's clear here that most of the data are here, and then some of them fall within this ellipsoid, and some fall within that ellipsoid. And so we use these ellipsoids as a kind of measure of the likelihood that something would happen. Well, you know, in the nucleosome, what you have is slide of around 2 angstroms and bending of around -18, somewhere around here. It costs 3 units of whatever this energy is to get there for CATG, but it's way more than 3 units to get there for ACGT. So that's the basis of how we're going to measure the likelihood of particular nucleotides assuming the kinds of states that you would see on the nucleosome. In fact, we see states in other proteins that aren't far away from what you would see at the most distorted states in nucleosomes. And then over here, this is just a kind of view looking at a CATG step or an ACGT step and showing you the differences. You can hardly see the bases that are, the C is in the background, and so the A and T is towards you, so it's darker. Whereas here, the A and T are in the background, and the C and G are in the foreground, and they're darker. But one of the things you can tell fairly easily is that the rings of these bases at the pyrimidine purine sites are not overlapping each other, whereas there are bits of overlap here. And this is an old idea that people have thought about for many years, that possibly it's favorable aromatic interactions between the aromatic rings, particularly when they stack above each other, that might stabilize arrangements, and so you won't have as much variation as in structures like pyrimidine purines, where you don't have that stabilization, and so you might get deformability. So this, of course, can go further to more detailed physical chemical studies to try to understand the underlying basis of that. So we are using that information, and here we've collected information for all 16 combinations of dimers on a sequence. So we have, 4 bases, so we can have an AA or a TT, an AG or a CT, and we've grouped them because, AA and TT are the same thing. Basically, GG and CC are the same nucleotides, almost. When you compute rigid body parameters, some of the values called tilt and shift change sign, but they have the same magnitude for these variables. And this is a color-coded image where we're looking at the central 60 base pairs of a nucleosome. So this is the dyad, 1 helical turn before, 1 helical turn afterwards, and so forth. And what we've done is we said, well, we have these kinds of bends, things like roll, very negative, slide, very positive at some points, and we compute what it costs with these derived energy functions for a CG to adopt whatever is happening at position -3 versus a TG or a CA or a TA or a GC. So red means it's very costly. So for example, here in the beginning, TA, CA, TG, CG can adopt what's ever going on, but AT, AC, GT, many of these others can't. And in fact, there's only a few points where it's very costly, red, for these pyrimidine purines to deform at least around the central 60 nucleotides of the base pair. And so, you could use this kind of information and say, well, where's the lowest one? Maybe this is the best one. Well, the darker blue it is, the better. So, maybe this is really good in this region, and this one's really good here, and so forth. And so you can try to deduce what sequences might prefer to bind to some particular structural template. Now, this is one of the best, this is the very best resolved crystal structures that was done in Tim Richmond's lab, 1.9 angstrom resolution. And here we just said, well, let's just take the structure that exists in the crystal, and let's just assign the numbers up to what the deformation energy is at particular points. And then let's take some generic DNA. And what I mean by generic DNA is we take the energies for all 16 steps at this point and average them. And so generic DNA has definitely something costly here, but it has, just all these numbers averaged. I don't have a map of generic DNA. But anyway, for generic DNA to wrap halfway around the nucleosome, and actually this is based on a symmetrized structure, it's very costly at 3 points, which are the points where there are those very large bends and shears in the region in contact with the H2A, H2B dimers. Whereas the same points on the structure that's crystallized are hardly costly at all. There are some points that are costly in the existing structures closer to the dyad. This is the beginning of the chain. This is 74 base pairs in, as opposed to the numbers we were talking before. This would be 0, -1, -2, and so forth. And then you can also, take these results where you have the total score, whether you have the DNA, where you consider the generic energy map or you consider the natural energy map, and you can see, well, what's the contribution of roll and what's the contribution of slide. And most of the cost that you observe for this particular structure is related to the cost of sliding at these steps. And so this is the threading experiment that we do, which is basically take a template, some structural template, and then, as I tried to illustrate, take a string, which is a string of nucleotides, and basically thread it like dental floss around here. And as it's threading, you'll notice I'm computing the score. So this is the natural sequence that's found in the crystal structure of the best resolved structure. And these are the central 60 base pairs of the structure. So we have a variety of ways that we can thread that longer 147 base pair sequence on the template in the center. And the one position that's a lot lower in this threading computer experiment is the one where the natural sequence is on the location that it is in the crystal. And then this is just the kind of energy function that you have. Those contour maps that we were looking at some optimized value that you expect for tilt or roll or twist or shift or slide or rise for a particular kind of nucleotide. And then we compare it with what is occurring at that nucleotide step. And then the function will allow us to look at, terms that might be the combination of roll and twist or roll and slide, because sometimes they're coupled. And then we have a force constant, which was basically related to where those contours were located. So the threading score is going to reflect which nucleotides are on the scaffold, and it's going to reflect whatever kind of function that's describing it. So these are 60 base pair steps, so it's the central 61 base pairs of that 147 base pair sequence. And then here is just one more example of this kind of threading experiment. We were looking at this result with the score where here this is threaded on a larger fragment, 129 base pairs, so there are fewer settings. And if we throw away roll, the score goes down, but there's still a dip. But if we throw away slide, that dip almost disappears. So all these things are basically saying that this sequence that's crystallized is favorable in that setting because whatever is happening that's costly to slide in general is involving nucleotides that can take up slide easily. And of course, those are pyrimidine, purine steps. And so you can play this experiment, with known positioning sequences, and you can compute the score, and you can compare it to where observed positions are found in kind of chemical cross-linking experiments. In this example, we found that the best resolved nucleosome structure fit very well. When we tried the best resolved, and then this is just telling us more about it. You have to have your template long enough. Your template can't be smooth. It doesn't fit as well. So there's technical things that I won't go into. When we were trying to study the 601 sequence and thread it on the best resolved nucleosome structure, we couldn't account for that data. But later, Victor found out that if he changed the nucleosome structure, there were certain nucleosomes that fit the 601 well. So all nucleosomes may look alike, but there are subtle differences. And in fact, that's probably why we started doing this collective information, because we wanted to understand, what might be some of the contributing factors to positioning. So these are 35 nucleosome structures that were available this past summer. There's a group of them that I'll call pure nucleosomes. None of them are really pure. Most of them have histones, which have the sequences that come from various species: frog, human, Drosophila, yeast, chicken. I think I might have them all, but I may have missed one. And then some of them are a little longer, some of them are a little shorter, some have different sequences, but I'll call these all pure nucleosomes, though in most of the cases, the DNA that's wrapped on them is the same old human alpha satellite sequence. Then there are 16 nucleosomes that, for obvious reasons, were studied because they have very interesting modifications. There are a bunch of things called syn mutants, where you'll have modifications of a particular amino acid in H3 or H4, and these kinds of modifications allow for processing of yeast without the SWE/SNF remodeling complex. There are examples where the histones are modified because these are site-specific mutations of methylation and such that are key to shutting off or turning on transcription. So there's a series of those. And then there are 7 structures with various kinds of ligands bound. Some are just things that fit in the groove, synthetic things designed in Peter Dervin's group. Some are anti-cancer drugs. Kurt Davey has done a lot of these kinds of studies. And then so here, what we're measuring is the distance between the phosphorus atoms. And this is a fairly reliable measure because some of them are very poorly resolved structures, worse than 3 angstrom resolution, 1 or 2 of them. And the best, there's only one of them that's 1.9. But this kind of measure is something that you probably put some confidence in. And the idea behind it is if you have a double helix, we know that the minor groove is going to get narrow when the DNA is touching the core of proteins and will get wider when it's away from it. So, these are mean values of the minor groove that, go from some number around 9 or something like that up to 15. And so you see this kind of variation. And then the color coding is the value that's observed in the particular structure. So we've color-coded this between 10 and 14. And that's because 12 is the mean value of all these data. And so if it's white, it means you're pretty much on the mean. And if it's red, it means that the minor groove is wider. And if it's blue, it means it's narrower. And so you can look at this image and you can say, oh yeah, they're mostly the same, but you see cute little jogs in here. And sometimes you'll see, for example, if you were to average over the whole structure, you could get the average minor groove width. So many of these things that have drugs have wider minor grooves than things that don't have drugs. And so you can pick out bits and pieces of information. Other kinds of things that you might see in here are little black dots. Like this one has 2 black dots. A lot of these have 1 black dot. And that's because some of the structures have different numbers of nucleotides. The ones that have 2 black dots have 145 nucleotides. The ones that have 1 have 146 nucleotides. And the ones where there's no black are 147. And these were aligned so that we got the best alignment of the minor grooves. And so you can see that where the differences are occurring are quite different places, whether you have the 146 base pairs versus the 145 base pair sequences. So if we didn't do that, these structures would look even more different. So we're talking about these as kind of phantom steps, which might be things that are going on in the real structure at these points to help bring these things in alignment. So, you know, at a global level, everything's looking the same. This is another way of looking at it. What we did is we picked out where the locations of the maxima and the minima were in different structures. And you can see them, you know, really sometimes jogging all over the place. Sometimes they're as much as 3 nucleotides apart, which means that the corresponding nucleotide, every time you have a nucleotide step, you're twisting 36 degrees. So 3 times 36 is 102 or something like that, or near 100. And so you're having what's on the inside, in some cases, changing very drastically. There's also examples where, you know, you can look at the structure and you'll find that, you know, there's extra maxima and minima, meaning that there is over or under twisting of the structure as well. This is what I mentioned about all these structures look the same. This sort of looks like an interesting carpet with some threads that are pulled incorrectly. Well, the incorrectly pulled threads are the structures that, as of last summer, were the only ones that had different sequences. And, you know, sometimes you'll see the same old sequence, but what's happening is something's missing here, or things are missing here, and so forth. And so, but also sometimes, even though there's just one little nucleotide that might be off, see here, you have some things that are off. You'll see changes that are happening very far away in the structure. This is the composition, A, T, G, and C, so you can see where the composition is different. And the one structure that's the best resolved one has the little arrows on it. So this is just to give you a sense. But now there are 3 or 4 more structures with other sequences, so we'll begin to learn more about those kinds of things. And then we were talking about these deformation scores. So again, the same 35 nucleosomes. Here, the alignment is such that we're sort of aligning the deformation scores. And so many times the alignments are, you know, where we're having to put these corrections are far away from where there are differences in the sequence. And then you can also see that there are certain steps, such as this one that's 1.5 turns away from the dyad, that is virtually costly in everything. And then you can see that there are some sequences. This one might be yeast, which is very costly. And this one might be the one with the cisplatin complexes that are very costly. And, you know, so you can begin to see some of these subtle kind of differences. I guess there was one other point I wanted to make here, is that with these max and min moving, what it means is that you're getting the DNA moving or wrapping around the nucleosome the same old way, but you know, there are different pathways. Actually, I have it better here. Yeah, I picked out these things called kink and slide steps, the ones where there's lots of bending and lots of shearing. And I picked out the ones that are the type that narrow the minor groove, have shearing in a positive sense, versus those that narrow the major groove and have the shearing in the opposite sense. And where are they located? And again, not every nucleosome follows exactly the right position. Sometimes you don't even have these steps. Sometimes the DNA is deforming smoothly. You're not having these sharp deformations. And then some structures, you know, may have more steps that are A-like than others. So again, this is just highlighting the fact that there's a lot of ways to get this overall fold of the structure. And it was these kind of subtle differences between the 147 and 146 base pair sequence that allowed us to thread 601 in one case and not in another. So one has to worry about deformability in the nucleosome. Okay, so then this is toward the end. we've been thinking about nucleosomes because we wanted to learn about the proteins differently. That is, and learn more about them so we could develop models. This is the plain old nucleosome. Here we represent it as a cylinder because we know where its superhelical axis is. And then what we do is we cut the cylinder at this point and open it up and look at the surface. So it's the idea of what you have with the globe, where we have the globe and then sometimes we can flatten out and get a flat map of the Earth. And so this is a map, schematic, of the DNA on the nucleosome. And this is a schematic of H3 and H4 and H2A and H2B with respect to the dyad, which is right here in the structure. So you see H2A and H2B are red and blue. They're near the ends. The DNA is this cyan stuff, and the H3, H4 are the green and purple. So it's just something we know. But then you can see interesting things. This is the vertical displacement of the helix. So sometimes this means that here's a tail that's coming up out of the structure from H3, for example. And then some of these things look funny because there are tails that are coming off of H2A that are fitting into the grooves of the DNA that are where H3 and H4 would be in the center. So you begin to see where things are if you think well with maps. And I always liked maps as a kid. So now we're thinking about proteins. Here is the radius. So we're taking our structure. And so this represents the center of the cylinder. This line represents the edge of the DNA. And in fact, you can see the DNA represented in terms of where we are along the surface of the structure and how far away we are from the center. And then color-coded in red are the nitrogen atoms that would carry a positive charge from lysine and arginine of the tails. And you can see them. And then colored in blue are the nitrogen atoms from arginine and lysine that are elsewhere in the structure. And colored in green, and I didn't know anything about histones, are a huge number of oxygens from glutamic acid and aspartic acid that are very closely associated with the nitrogen atoms of arginine and lysine. I mean, I only know a little bit about proteins, and so I was familiar with things like Barry Honig making a big deal about ion pairs in the middle of proteins, but usually there was one of them. And I never expected, because I didn't know anything, that there would be so many. And so what you can do is you can, map these things versus histone type. But here we're just counting them, and we're saying, how far are we from the center? And let's count positive charges. And they build up in the center, and then they build up as you go through the DNA, and then they build up even more as you count them. And these are the ones that are in the histone tails. If you count the negative charges, they basically build up in parallel with the positive charges in the center, build up a little bit into the DNA, and then level off. And so we have a neutral bit, and then we have this charge building up through the DNA and building up through histones. And so when we're developing our model, we thought, well, we could save a lot of time if we didn't have to worry about these charges in the middle. And so we counted, how many of these charge-bearing atoms were near each other, and it seemed like a reasonable approximation to just not worry about them. And so we'll worry about clusters of charges on the DNA, representing the sugar phosphate backbone, and we'll represent clusters of charges from the histones in that region of buildup around the DNA, and we'll worry about clusters of charges in the histone tails. And so this is the basic element. If we're going to be studying multinucleosomes, we're going to have DNA that's connecting nucleosomes. That's pretty much like generic elastic DNA. When nucleosomes are there, we're going to have these charges, and we're going to keep account of them. And so we've done a lot of computer simulations, and this is representative of the kind of thing that we've done. Here we have about 13 nucleosomes simply represented by their wrapping around the histone core. You're not seeing them. And we did simulations where we considered all those charges that we were talking about. And then we did simulations where we threw away those charges on the tails, just a small part of the charges on the system. And these are snapshots of the kind of pictures that you get, illustrating that many times the tail-containing simulated fibers have more bending than the tailless. This makes it more quantitative. We can compute the distances between the ends of chains. These are chains that have 13 nucleosomes spaced 177 base pairs apart. So it's, 2,500 or so base pairs long. And this is the distribution of the end-to-end, where the ends are. And so this number measures the likelihood that the ends will be there. The area under this curve is 1, so that's all configurations that you see. And so basically at shorter distances, we have a greater likelihood when we have these tails in our simulations for the ends to come together than when we don't. And if we take DNA that's the same length, but it doesn't have nucleosomes bound to it, first of all, this number goes from 0 to 0.001, which is right in that regime. And the buildup is between 150 and 600, way into outside the building probably. And so you can see that there's a great enhancement in the likelihood of the ends coming together when the nucleosomes are there, and it's even greater with the tails there. So we did a little bit more just to look at this kind of thing. This is an example where we have 25 nucleosomes, and they correspond to column 1, 2, 3, 4, 5. And then these little bigger than a pixel, represent colors that tell you in 4,000 representative configurations how many nucleosomes are at position 1, 2, 3, 4. So very dark blue means there's nothing. So there's a lot of things with nothing, or there are no contacts of that nucleosome with anything else. And then toward the middle, most of the colors you see are maybe light blue, maybe some green. So it means that in these configurations, most of the time you'll see around 2. And these are averages of those numbers. So the 3 nucleosomes on the ends don't make as many contacts as the ones in the middle. And on average, in 4,000 simulated states where there's nothing bumping into each other, you get close to 2. But if you have the tails there, the colors are very different. You see a lot of yellow and even some orange. And in fact, the number is a little bit higher. Actually, I think I forgot the number, sorry, when I made that, because this is a different scale. And so there are definitely more interactions with this kind of thing. So just the removal of a little bit of charge is changing the distribution function, is changing the contacts, and then this is one final one that's telling you the types of contacts it is. This one has 50 nucleosomes. So here's nucleosome 1 to 50, here's nucleosome 1 to 50, and then it's color-coded. So we're looking at what's the likelihood that nucleosome 50 interacts with 49, or the likelihood that nucleosome 50 interacts with 30, or with 15. And this is the negative log of the probability. So red means there's more likelihood, and blue means there's less likelihood. And this is for tailless, and this is with tails. And so again, you see numbers that aren't as blue, which means there's more likelihood of, like 20 interacting with 45 than 20 would interact with 45 in the negative tails. So there's interesting implications for communication. And this is the experiment that Vasily's doing, which is he's set up a system where he has a binding site for an enhancer protein, in his case the NTRC activator protein, and he has the binding site for RNA polymerase and the sigma factor. And then he has genes that are transcribed. And so he's measuring the likelihood that a protein at this end is interacting in a protein with that end by measuring whether or not he gets gene product. And then in between is either plain old DNA or nucleosomal DNA. And right now we're to the point of trying to begin to compare our computed values with his measured values. And so with that, there were 3 messages that I wanted to give you today. First was, looking at things differently. And in the case of the nucleosome, we came across accidentally something about the displacement of the base pairs being very important, and finding out that nucleosomes and crystals took these up at pyrimidine purine sites, and that if we used derived information from crystals that took advantage of this information, we could account fairly well for the positioning of some known positioning sequences based on what's known in high-resolution structures. And then we learned, how there are some subtle differences among structures that might be useful in understanding positioning. And then now we're learning some things about histone ions. And in this case, we're learning about the ions in the tails and how they're involved in long-range communication. And with that, I'll end, and thank you for your attention.
So thanks. Questions for Wilma, although take the liberty of starting, which is, so for some DNA sequence of your choice, there'll be a favored location along the DNA, and that will have some nice negative energy. And then if you move half a helical turn away, that'll be a worst possible energy. And I'm interested to know, what's your thought about the free energy difference between the valley and the peak?
Okay, well,
in other words, how rough is the DNA landscape?
You're always asking me, why don't you put kilocalories per mole to your numbers?
That's really what I'm asking.
And I'm always saying, John, I have to be very careful, because people will think I'm crazy if I put these numbers there. And so it's basically that debate. I think.
But among us friends, what's the answer?
But among friends, as you collect more data, and I, try to gather information, I may be able to do that, because some of the things we can do, we can take these data and we can make DNA sequences, and we can compare them to things like the persistence length. And we can see what sort of apparent temperature we have to add to our system so that we could reproduce the persistence length. Or another way, how do we scale this to kcals per mole? And some of the data that we've taken from protein DNA complexes, this correction factor is close to 1, so that those numbers 1, 2, and 3 might be 1, 2, and 3 kcals.
Yeah, okay. And then also I was very struck in the steric diagrams for the pyrimidine purine and then the purine pyrimidine steps. Even in the purine pyrimidine step, there was almost no base stacking. I was kind of shocked by that.
But a lot of people, I was emphasizing the rings.
Yeah.
But there's old stuff that goes back to Charlie Bug, Sunderlingam, and others. And they also try to emphasize the fact that images like that are not telling you a lot about the exocyclic carbonyls and the exocyclic amino groups, which are just as much a part of the interaction. And so you will see, and Charlie Bug pointed this out, I don't know, 1970, ages ago, that there are really interactions of these exocyclic groups with the rings that are most important. And if you were to be doing high-level quantum calculations, if you left those off, I think you would see, major differences.
I see. Okay. Thanks. Yeah, Tom.
Yeah.
So you kind of pinpointed the slide as one of the more useful predictive aspects in figuring out these bends, and those deviations are about an angstrom when they're significant.
In the most deformed states, in the best resolved structure, that is 2 angstroms, which is a huge number. It's the kind of number that, if repeated, all the base pairs are displaced from the center of the helical axis. But you do see examples in other structures where, you're moving in that direction. And this is this form called cDNA that hasn't gotten a lot of attention. In the DNA plain old vanilla DNA high-resolution structures, Dickerson introduced the idea of what he called B1 and B2 DNA, and that may be in the textbooks. And B2 DNA is this highly sheared state.
Sure. So, and you identified, you looked for these because you saw these at Kink sites or at
Right.
particular events.
We looked at them because David Mccandlish had this data, and all of a sudden we saw a superhelix become a figure. And then Victor and I thought, how do we present it and make it interesting to people? Well, tell them that we should look at nucleosomes differently, but it was all accidental. So that first slide came long after we made the movie.
So did you look for other cases of slide outside of the bending regions and did that?
We've looked at these, and for example, we can collect data on all kinds of structures. And just like if I move my middle finger, my last finger, my next to last finger moves, they're coupled. Well, in the case of nucleotides, these rigid body parameters are just artificial things. It's just easy physical parameters that you would use in physics. But there's chemistry in between them, and basically every time you have slide, you have bending and twisting. They all go together like two fingers moving together. So
more linear parts.
right. So there are proteins, like TC3 resolvase that have whole stretches of A-like steps of DNA in them. And they have the slide, the bending, and the untwisting at the same time. So slide usually doesn't happen by itself. But then there's cute computer games you can play, because you can imagine how you can pull the DNA off the cylinder by just having small series of translations. And you don't have to stretch it in the sense of bending it. A lot of people, when they're thinking about how the DNA moves as a deformation or a bubble moves through the DNA, they sort of imagine, well, something comes out and it bends. But you could just pull the DNA out to do that and pull it out sequentially. So it just gives you a different way of thinking about things.
Okay, well, let's thank Wilma again.